Very good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and yes, we are in our second session for the chapter Photosynthesis and Higher Plants. In our previous class, we have dealt more clearly about early discoveries, chloroplast cell organelle, action spectra, absorption spectra, chlorophyll pigments, photosystems in brief. So, in today's session, we are going to clearly visualize cyclic and non-cyclic photophosphorylation and Chemi-Osmotic Hypothesis. So, yes, stay tuned till the end of the class beta. So, first let us recall the gist of photosynthesis in detail. Photosynthesis, we have light reactions and dark reactions. Light reactions also said to be light dependent reactions which come under photochemical phase. Okay, they come under photochemical phase, because in this process the photons, that is light energy, is being converted into the chemical energy. This is photochemical phase. The light energy, that is photons, is being converted to the chemical energy, that is ATP and NADPH. Most importantly, that we have to focus is the sites or locations of the reactions. First, if you have to take beta, the very first step that happens is absorption of light in the form of photons. Absorption of light in the form of photons happens in photosystems. Photosystems which are located in thylakoid lamellae. They are located in thylakoid lamellae. And the second one is photolysis of water. Utilizing the energy of photons, water whichever is present in the thylakoid lumen is photolyzed or hydrolyzed to 2 protons, 2 electrons and an oxygen atom. Okay. So, here the second step is photolysis of water which occurs in thylakoid lumen, and then the light energy is being converted to the chemical energy, that is assimilatory power is formed, ATP and NADPH, in the stroma of the chloroplast. In the stroma of the chloroplast. This assimilatory power, you can say products of light reaction are utilized, the products of light reaction are utilized for fixation of carbon dioxide. You can say this as carbon dioxide fixation or carbon dioxide reduction or carbon assimilation, where carbon dioxide is fixed to the complex glucose molecule utilizing the assimilatory power, that is ATP and NADPH. As this process is not directly dependent on light, but it is dependent on the products of the light reaction, this is said to be light independent reactions. Calling light independent reactions light independent reactions as dark reactions is a misnomer, but then because dark reaction does not mean this takes place during night time. Okay, this just means light independent reactions. This is not directly dependent on light, these are dependent on the products of light reaction. And as this is involved in the synthesis of biomacromolecules, this is also said to be biosynthetic phase. Biosynthetic phase. Fine. So, yes, light reaction also called as photochemical phase, dark reaction is also called as biosynthetic phase. Clear, right, ma? So, this is the gist of photosynthesis that we have discussed. So, till here the process is very clear to us. Now, let us focus on how the assimilatory power is formed, how ATP and NADPH is formed, and how the dark reaction is being taking place. C3, C4 and CAM cycles we will be looking into the next class, but this formation of assimilatory power we will be looking into now. Okay. Tanveer, very good evening beta. Continue with the class. Any doubt anywhere you can just ask me, ma. So, yes. So, before learning into the photophosphorylation concept, you should be very clear about oxygenic and anoxygenic photosynthesis. So, based on whether oxygen is evolved at the end of photosynthesis or not, we have 2 types of photosynthesis. What are they? Whether oxygen is evolved or not, we have 2 types of photosynthesis, oxygenic photosynthesis and anoxygenic photosynthesis. Okay, oxygenic photosynthesis and anoxygenic photosynthesis. Oxygenic photosynthesis takes place in higher plants where oxygen is evolved. So, in them non-cyclic photophosphorylation occurs. Non-cyclic photophosphorylation. Whereas anoxygenic photosynthesis, which is also called as bacterial photosynthesis, cyclic photophosphorylation occurs mostly. Cyclic photophosphorylation. First, to know about non-cyclic photophosphorylation and cyclic photophosphorylation, no issues beta, you can continue ma. Yeah. So, before knowing this non-cyclic and cyclic photophosphorylation, you should be clear with the term of photophosphorylation. Photophosphorylation. Okay. What is photophosphorylation? Photons, that is light energy, it is utilized for phosphorylation, to addition of phosphate group, wherein stroma ADP is added with inorganic phosphate utilizing the light energy, and that is how ATP is formed. So, formation of ATP by utilizing the light energy is called as photophosphorylation. So, this photophosphorylation is of two types, that is cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation occurs mostly in anoxygenic photosynthesis or bacterial photosynthesis, wherein stroma lamellae is involved. This takes place in stroma lamellae. Fine. Whereas non-cyclic photophosphorylation occurs in oxygenic photosynthesis, that is in higher plants. This occurs in thylakoid lamellae. Thylakoid lamellae. Till here it is clear, right beta? So, look at here. First, light which is absorbed as photons in non-cyclic in cyclic photophosphorylation, that is anoxygenic photosynthesis, stroma lamellae is present and there is only one photosystem, that is PS1, whereas here two photosystems are involved, both PS1 and PS2. Okay, two photosystems are involved over here. So, first let us finish the discussion of cyclic photophosphorylation. Once again, cyclic photophosphorylation mostly occurs in bacterial photosynthesis. Tanvir, I will be answering your doubt later beta. First you focus on the cyclic photophosphorylation, okay. So, this occurs in bacterial photosynthesis, cyclic photophosphorylation, stroma lamellae, inside stroma lamellae only photosystem 1 is present. So, this photosystem where the photons are being absorbed by accessory antenna and the core pigment is present, this light energy is being excited wherein two electrons are given to ferrous sulfur complex or FRS complex, or you can directly say to ferredoxin. Electron transfer is first to ferredoxin, then cytochrome B6. In the process of transferring the electrons from ferredoxin to cytochrome B6 and from cytochrome B6 to cytochrome F, ATP formation takes place, okay. And then last electron acceptor, or the ultimate electron acceptor in cyclic photophosphorylation is plastocyanin, and then the electrons will be returned back to the photosystems. As it is in the cyclic manner, this is called as cyclic photophosphorylation. So, this cyclic photophosphorylation which occurs in the stroma lamellae photosystem, absorbing the light energy, electrons get excited where the primary electron acceptor will be ferredoxin, then cytochrome B6 and then cytochrome F, wherein ATP molecules are formed, and then plastocyanin, that electrons will be again submitted to photosystem 1. This is the process of cyclic photophosphorylation. I will give you 2 minutes of time, just verify it clearly. Any doubt anywhere you can ask me beta. So, Tanvir, both red light and blue light, they in the presence of both red light and blue light, maximum amount of photosynthesis is conducted. Okay, that is proved by E.W. Engelmann's experiment. And when you say about the plants, when you are keeping indoor or outside, it is not the direct sunlight that is passing to the plants. Many reflections of the sunlight, many rays of the sunlight of different wavelengths will be landing onto the photosystems which is present in the mesophyll cells. Okay. So, the light energy which is being taken, mostly red region and green region of the light will be absorbed to the maximum, and green light will be reflected. That is how the leaves will be green in color. Okay, as green light is reflected, absorbing blue and red light. Okay, understood beta? Yeah. So now I think it is clear for you about cyclic photophosphorylation. Now let us understand in detail about non-cyclic photophosphorylation. Okay, yeah, non-cyclic photophosphorylation. This is also called as Z scheme. Okay, non-cyclic photophosphorylation, also called as Z scheme. To understand this better, okay, look at here. First, let us assume there is a leaf which is composed of mesophyll cells. Each mesophyll cell will be having like 40 to 50 chloroplast organelles. Fine. So let us assume this as a chloroplast organelle which is double membrane bound. Each chloroplast will have membranous sac-like structures. This membranous sac-like structure is thylakoid. Stalk of this membranous sac-like structure is grana. One grana is connected to other grana by stroma lamellae. Right ma? One grana connected to other grana by stroma lamellae. We have learned this thylakoid are the membranous sacs which are bounded by thylakoid membrane. Okay, these are bounded by what? Thylakoid membrane. Clear, right ma? So here if I have to consider this as one thylakoid and this is the thylakoid membrane, this is thylakoid lumen. Okay, this is thylakoid lumen and this is thylakoid membrane. So look at here carefully. Yeah. So thylakoid membrane and lumen is clear to you, right? We are taking the small portion because photosystems are present in the thylakoid. Okay. Very good, Shiva. Just continue with the class. Yeah. So here if you see thylakoid membrane and lumen. Let us suppose this is the thylakoid lumen and this is the thylakoid membrane. Clear, right now? In a leaf mesophyll cell, it may have 40 to 50 chloroplast organelles. One chloroplast organelle, let us assume, each chloroplast will contain what? Membranous sac-like structures, thylakoid membrane. One thylakoid connected to the other by stroma lamellae. So let us assume the thylakoid membrane, this is the thylakoid lumen and this is the membrane. And inside thylakoid membrane, we know what are present? Photosystems are present. Yes or no, ma? Okay, photosystem 1 and photosystem 2. So now utilizing the light energy, that is photons, okay, photolysis of water happens by oxygen evolving complex which is attached to photosystem 2, okay, photosystem 2 and inner membrane of the thylakoid. Attaching to photosystem 2 and inner membrane of thylakoid, there is oxygen evolving complex. This oxygen evolving complex will mediate the photolysis of water, where water molecule is being photolysed to 2 protons, 2 electrons and an oxygen atom. Clear, right? So here protons get accumulated in the thylakoid lumen. Remember this point, very important. Protons get accumulated in the thylakoid lumen. Electrons will get excited. Photons somehow will transmit the electrons to the reaction center. That electrons will be transmitted uninterruptedly by this phenomena where the photolysis of water is taking place inside the lumen. Clear, right? Now, so here if you see the electrons being excited to the reaction center, this reaction center is chlorophyll A. Okay, chlorophyll A is the reaction center. and from here the primary electron acceptor is pheophytin. Okay, what is the primary electron acceptor beta? Pheophytin is the primary electron acceptor. From this the electrons are being accepted, 2 electrons are being accepted by plastoquinone. Plastoquinone accepting the electrons from pheophytin, parallelly it accepts protons also. Pheophytin is on the outer side of the thylakoid membrane, plastoquinone will be towards the inner side. Okay, accepting the electrons, it will parallelly receive the protons from the stroma. This is the second most important point, remember. Okay, first is accumulation of protons in thylakoid lumen, second is protons are being pumped from where? Protons are being taken from the stroma, and this plastoquinone will be converted to plastohydroquinone, where protons are deposited into the lumen and electrons are being given to cytochrome B6F. From cytochrome B6F, the ultimate electron acceptor of photosystem 1 is plastocyanin. Okay, once again let me give you a quick recap of this. From the oxygen evolving complex, the photons, light energy it will be releasing. Oxygen evolving complex is attached to the photosystem 2 and inner membrane of the thylakoid, wherein water molecule is photolyzed to 2 protons, 2 electrons and an oxygen atom. This electrons will be pumped or served as a continuous supply of electrons uninterruptedly to the reaction center and to the protein molecules in the photosystem 2. From the reaction center, primarily the electrons are accepted by pheophytin. Pheophytin is the primary electron acceptor of photosystem 2, or pheophytin is the primary electron acceptor of non-cyclic photophosphorylation, remember that. From pheophytin, then 2 electrons are being given to plastoquinone, then plastohydroquinone. Parallelly this takes electrons and protons from the stroma, and then electrons are submitted to cytochrome B6F, and the ultimate electron acceptor from photosystem 2 in non-cyclic photophosphorylation. If that is the question, your answer should be plastocyanin. Clear, right beta? Now the electrons are being pumped from plastocyanin to, okay, electrons are being pumped from plastocyanin to ferredoxin. Okay, ferredoxin. Ferredoxin is the primary electron acceptor in photosystem 1. Here the reaction center, that is chlorophyll A, maximum wavelength of this reaction center is P680, 680 nanometers. So we can say it as P680. Maximum wavelength of the reaction center in photosystem 1 is 700 nanometers. We can say it as P700. Clear, right? Now ferredoxin will give 2 electrons, okay, 2 electrons to ferrous sulfur complex. As such, the ultimate electron acceptor from ferrous sulfur complex is NADP+. Okay. Yes. So the ultimate electron acceptor is NADP+. NADP+ is present towards the stroma side of the outer thylakoid membrane. Okay. Till here all the protein molecules in photosystem 1 are present inside the thylakoid membrane, inside the photosystem 1. But NADP+ is present towards the stroma side of outer thylakoid membrane, which is the ultimate or the final electron acceptor in non-cyclic photophosphorylation. Accepting 2 electrons, this NADP+ will be reduced to NADPH plus H plus. Okay, it will be reduced to what ma? NADPH plus H plus by a enzyme, that is, this is an enzyme mediated process. That enzyme is NADP reductase. Okay, the enzyme is what ma? NADP reductase. Okay, so till here it is clear to you, right? Fine. Once again, let me give you a quick recap of non-cyclic photophosphorylation, and let me give you a clear idea on uphill and downhill movement of the electrons. So here if you see, first the electrons from the oxygen evolving complex will be excited towards uphill when it is giving to the primary electron acceptor, that is pheophytin. From here electrons will be moved in the downhill again. okay, downhill till the end of the last electron acceptor of photosystem 2, that is plastocyanin. From here again the electrons will be excited in the uphill movement to ferredoxin, and then again downhill to the ultimate electron acceptor of non-cyclic photophosphorylation, that is NADP plus. Till here it is clear to you, Nama? So here any doubt anywhere in the non-cyclic photophosphorylation, you can just ask me. What you have to highlight and focus in this non-cyclic photophosphorylation? Which is the primary electron acceptor, which is the ultimate electron acceptor, last electron acceptor of non-cyclic photophosphorylation, primary electron acceptor of photosystem 2, what is the reaction center, what is the protein molecules flow in which the electrons are being accepted, okay, which molecule is towards the outer side of the membrane, which molecule is towards the inner side of the membrane. This and all should be highlighted when you are going through non-cyclic photophosphorylation. Okay, so this is the complete concept that we have covered. Any doubt anywhere, you can ask me, ma. Okay, fine. Yes. Explain P700. Yeah, P700 is nothing but beta. See, this is photosystem 1. In photosystem 1, the reaction center is again chlorophyll A. Okay, the reaction center is again chlorophyll A, but the chlorophyll A which is present in the reaction center of photosystem 2, maximum wavelength is 680 nanometers. So, it is P680, but here P700 which is present in the photosystem 1, the maximum wavelength is P700, clear, right? So, maximum wavelength of the reaction center which is present in photosystem 1 is 700 nanometers, which is also said as P700. Okay, Tanvir, any other doubt, ma? Yes. So, if non-cyclic photophosphorylation is clear to you, here I would like to tell you one point, that see, the assimilatory power formation is what? ATP and NADPH, and we said that photophosphorylation is nothing but formation of ATP utilizing the energy of photons, combining ADP and inorganic phosphate, ATP should be formed. But then in non-cyclic photophosphorylation, at the end of non-cyclic photophosphorylation, only NADPH is formed, ATP hasn't formed yet. So, how ATP is formed is being explained in Chemi-Osmotic Hypothesis. Before learning Chemi-Osmotic Hypothesis, once look at this diagram again, okay, look at this diagram, okay, carefully. So, can you see, this is what? Thylakoid lumen, and this is thylakoid membrane, this is chloroplast stroma. So, here photosystem 2, maximum wavelength P680 of the reaction center, light is being passed out, okay, and the ultimate and the continuous supply of electrons are being given by this oxygen evolving complex, where the electrons are primarily given to pheophytin and then plastoquinone. Plastoquinone parallelly accepting the protons, turning out to plastohydroquinone, where hydrogen atoms or protons are being pumped to the lumen and electrons to cytochrome B6F, okay, then to plastocyanin. Plastocyanin, can you see here beta, which is located on the inner side of the thylakoid membrane, okay. Plastocyanin to again photosystem 1, where ferredoxin is towards the outer side of the thylakoid membrane, and NADP plus, which is the ultimate electron acceptor, is present in the stroma, okay. And this is an enzyme mediated process where NADP reductase is the enzyme that reduces NADP plus to NADPH, okay. And then from here, how ATP is being formed is explained by Chemi-Osmotic Hypothesis, okay. This is being explained by what? Chemi-Osmotic Hypothesis. So, before learning Chemi-Osmotic Hypothesis, once let us clearly look into the differences between cyclic and non-cyclic photophosphorylation. So, here this is the diagrammatic representation of cyclic and non-cyclic photophosphorylation, and here you can see the differences keenly. First, cyclic photophosphorylation is nothing but what, ma? Synthesis of ATP during the light reaction of photosynthesis by cyclic passage of electrons to form P700. When plants need more ATP than NADPH, then cyclic photophosphorylation is operated even in higher plants. But mostly this occurs in the bacterial photosynthesis. Whenever plant requires more ATP than NADPH, then cyclic photophosphorylation will be operated in this too. And then non-cyclic photophosphorylation is nothing but synthesis of ATP during the light reaction of photosynthesis, in which electron donor is required and oxygen is produced as a byproduct. This occurs in isolated chloroplast and photosynthetic bacteria, and this occurs in the higher plants, algae and cyanobacteria. In cyanobacteria also oxygenic photosynthesis only takes place. This occurs in anoxygenic photosynthesis, this occurs in oxygenic photosynthesis. Here final electron acceptor is P700, here it is NADP plus. Remember, in non-cyclic the ultimate electron acceptor is NADP plus. And then electrons move in cyclic pattern. Okay, electrons will be moving in the cyclic pattern. Here electrons will move in a line or a pattern, which is also called as Z scheme. Okay, here only photosystem 1 is involved, here both photosystem 1 and photosystem 2 will be involved. Electrons are first expelled from reaction center of photosystem 1. Here electrons are first expelled from the reaction center of photosystem 2. All the differences should be highlighted beta, because whenever statement based questions are given, assertion reason questions are given, if you are keenly noted with the topic, then you can easily answer. Okay. So, and then electrons return to the P700 and passing through electron transport system. Electrons return to P680 here in non-cyclic photophosphorylation after photolysis of water. Understood? And then Chemi-Osmotic Hypothesis. So, till the end of non-cyclic photophosphorylation, if you are having any doubt anywhere, just drop a message in the chat box. I will be answering your doubt. Okay? Just give me 2 minutes. Yes, now Chemi-Osmotic Hypothesis. So, at the end of non-cyclic photophosphorylation, we got to know how NADPH is formed. Now, how ATP is being produced by this electron gradient is being explained in the Chemi-Osmotic Hypothesis. So, Chemi-Osmotic Hypothesis is first proposed by Peter Mitchell. It is proposed by Peter Mitchell. So, here if you see, just a second, yeah, Chemi-Osmotic Hypothesis, which is given by Peter Mitchell, wherein, see, there are 3 points to be highlighted. Okay, 3 points to be highlighted. Okay, which is given in your NCERT as a very big in one complete page it is given, which explains about Chemi-Osmotic Hypothesis. Okay, the very first thing is photolysis of water will bring the accumulation of protons. Protons get accumulated in the lumen because of photolysis of water. Okay, H plus accumulates in lumen. Okay, second is plastoquinone accepting the protons turns to PQH2 and protons from stroma are pumped to the lumen again. Okay, protons are pumped from stroma to lumen, by what? PQH2. And third most important point is whichever minimal protons are present in the stroma are being taken by NADP plus and they are converted to what? NADPH. So, whichever minimal protons are there, that NADP plus is converted to NADPH, which is reduced to NADPH. So, this generates more proton accumulation in the lumen and less protons in the stroma, which generates a proton gradient between lumen and the stroma. Okay, this generates proton gradient between lumen and stroma. Okay. So, this proton gradient which is generated between the lumen and stroma, where a pump is activated, that is called as F0 F1 pump or ATP synthase. What is it, ma? ATP synthase or F0 F1 pump. Fine. Wherein F0 is embedded inside the thylakoid membrane and F1 is towards the stroma, where in a single run it will pump 4 protons which will form one ATP molecule. ADP and inorganic phosphate are present in the stroma and ATP molecule will be formed. Okay. This is the complete thing about chemi-osmotic hypothesis. So, here you can see the brief description of ATP synthase. ATP synthase consists of the following components, that are 2 parts, one is F0 and CF1. F0 is embedded inside the thylakoid membrane and this forms a transmembrane channel that carries facilitated diffusion of protons from lumen to the stroma. F1 will protrude on the outer side of the thylakoid membrane which faces towards the stroma. Okay. That breakdown of gradient will provide enough energy to cause the conformational change in the CF0 complex which is placed inside the stroma. This makes the enzyme synthesized, that is ATP, and this complex is called as what? F0 F1 complex. As a channel is involved to pump the protons from lumen to the stroma, this is called as facilitated diffusion. This is an example of facilitated diffusion. Facilitated diffusion. Clear, right? And then, okay. So, with this we have finished out the cyclic photophosphorylation, non-cyclic photophosphorylation and the ATP synthesis, that is chemi-osmotic hypothesis. In our next class of this evening batch, we will be dealing completely with the dark reactions, that is C3, C4 and CAM cycle. Along with that, we will be also looking into the factors affecting photosynthesis. So, with the next class, we will be finishing off this chapter, that is photosynthesis in higher plants. To learn the objective from this chapter, you have to watch the playlist of the questionnaire that I am doing, wherein all the NEET previous year questions of this chapter and the advanced questionnaire of this chapter are covered. Okay. So, stay tuned, keep learning, keep growing. Thank you.